So good day, everyone. I'm just going to present what I've been doing at DSI since now 2016, so part of my master and my PhD, and I will just present a little bit what's my future project as a postdoc here at DSI. So I worked mainly on therapsids, and I worked on their ontogeny, their taxonomy, and also their phylogeny. So first of all, who are the therapsida? Therapsida, they are clade of basal synapsids that evolved from a pelycosaur grade and eventually gave birth to us, the mammals. They appeared during what we call Holson gap, which is a fossilization hiatus or an extinction event that happened between the early Permian to the middle Permian. And at that time, during that time, we have absolutely no fossils. So we have no record of early evolution of those animals. And that gap is actually defined by the transition from pelycosaurs, from geological layers where you can find most of the pelycosaurs mostly in North America, to layers, geological layers where you can find therapsids, mainly in South Africa, Russia, and China. The problem with those animals, it's like already at the base of the fossil record in South Africa, they show like a very high diversity. So we have absolutely no idea of what happened at the beginning of the evolution. They are made of 6 groups. There's the Biarmosuchia, the Dinocephalia, Anomodonts, Pterocephalians, Gorgonopsians, Pterocephalians, and then Cynodonts, which are the ones who gave birth to the mammals later on. I'm just going to go through every group and present them to you. So Biarmosuchian, they consider to be one of the basal-most therapsid for now. They are all carnivorous and develop like very large canine. They develop cranial bosses and ornamentation on the skull and very thick pachyostoses skull. And some of the species might have been also nocturnal, like according to some studies who have studies like the size of the eyes compared to the size of the skull. Then there's Dinocephalian. The basal-most Dinocephalians are the Onteosauria. They consider to be carnivorous, and they also develop thick pachyostoses during their ontogeny, while you have later groups in evolution that are like mainly herbivorous with like crushing heels like teeth. And those ones, they are believed that they might have performed like headbutting, like nowadays sheep also, according to some studies on like the inclination of their skulls. But that's what my student Tristan is doing now, so you can ask any question about it to him. And here I'm co-supervising him with Julian now. Anomodonts. So anomodonts, they were mainly herbivorous. And with like the cynodonts that develop like what we call a tiny tusk there, and maybe a beak for some of the species, there's a huge fossil record for them. So they are one of the perfect examples that you can use for paleobiology studies and study for diversity and so on. They show that they have 2 radiation. They undergo 2 major radiation, once during the middle Permian, then they go through the PTB. They somewhat survive the PTB to undergo a second major radiation during the early Triassic. Some of those species, like Sumenia, that Russian species, might have been living in tree and arboreal species, while others, like this one, Caringasaurus, might have developed like underground way of living, according to some studies on the inner ears, which show that that species might have been like doing some burrows. We have then Gorgonopsians that were the carnivorous apex predator of the time. Some of those species display like a double canine morphology, which could be either linked to ontogeny or even linked to like just like replacement canine through the life of those animals. They had like very large canine with like serration, showing like a carnivorous way of living. Then we have Pterocephalians. Pterocephalians seems to have like Gorgonopsians. They were also carnivorous with like a pattern of double canyon for some of the species, which it was actually back in like the 60s to 70s. That character here was one of the synapomorphic for basal Pterocephalians, but now we have some studies like the one from Luke Norton and his PhD showing that that was more related to canine replacement through the life of the animal. They are characterized by an enlarged temporal fenestra and they have a reduced temporal portion, and some of them have like a parietal crest that is similar to also cynodonts. And some specimens might have shown like what we call turbinoles in the nasal, which could show like a sign of early development of ontogeny, but that I will invite you to ask Valentin at his PA postdoc project. While most studies show that they were monophyletic, there's some suggestion like from Jennifer, like in 2007, that says that maybe they might be paraphyletic, with like some of those pterocephalian basal ones branching with the cynodonts, actually at the base of the cynodonts there. I will talk about that a bit later during the talk. Finally, cynodonts, the one that gave birth to the mammals, like through the evolution of therapsids, they show that they are more and more looking like mammals. So they were mainly carnivorous for some species that were not. Those specimens tend to lose their binaural foramen, like we have in mammals today. They lose their lumbar ribs, like again, they evolving more and more towards like mammal-like morphology. And they also develop a secondary bony palate inside the nose. Some of those species, actually all of the species that survive the PTB, they will later on develop what we call the fully erect position, while the Permian species have more like what we say half erect positions there. So that was the introduction about what are the therapsids, like showing to you like the variety of morphologies and species that we are. I'm just going to divide my talk in 3 parts. So the first part, I'm going to talk about ontogeny of those animals, and I'm going to take a case studies and on Bjarnesukians. While the second part, I'm going to talk about taxonomy, how we do taxonomy in therapsids, and I'm going to take like an example I had on basal and amadons. And finally, speaking of the early evolution of therapsids, there's still some questions. So during my PhD, I was able to perform some phylogenetic analysis that I'm going to present at the end of the talk. During those 3 parts of my PhD, I used various like methods, mainly phylogenetic methods, but also CT scan methods. All of my specimens were scanned, and I was studying like the 3D reconstruction I had of them. So CT scan, it's the use of a high beam energy in order to perform a 3D analysis, a 3D X-ray of your specimens that enables like really nicely details to obtain nicely detailed morphology. So there's 2 ways that you can do a CT scan. So you even go for microtomography, where you have directly like the source of light that's going onto the specimen that is rotating on the platter. And then at the back, you will have like the camera acquisition. At the end, through the acquisition, you can obtain like a 3D X-ray of your specimen, which then enables you to do 3D reconstruction. Or you can do the synchrotron way, especially if you want very high details of your scans, so that with the difference on synchrotron, it doesn't give you directly the light energy. It has to produce the light energy. So the light energy is produced through the acceleration of particles inside the inner circle. On the one from the synchrotron, in France, in Grenoble, the one that you use, it's actually electrons that are accelerated through the circles, and through a series of magnets, those electrons are going to be excited and produce a high beamline energy. That beamline that is produced, that you can use for like performing like CT scan and 3D scans on your specimen, which is actually, it's kind of like a microtomography, but like in bigger scales. So you would place your specimen in the middle of the room with like the source of light that is here, and then it goes through your specimens, and then you have like a bigger camera acquisition at the back. So it's basically a microtomograph, but like much more bigger scale to have a higher resolution. Whether you go from like microtomography or synchrotron, you obtain, in both cases, you obtain like a 3D radiography of your specimen that you have to then study using 3D software. In my case, I used Aviso. And for a lot of us using 3D, like after a really long night and a lot of energy drink and coffee, you can obtain your 3D model of your specimen through segmentation. During my PhD, I was doing manual segmentation. So I would like segment every 10 slides and then interpolate in between the slides. The last part of my talk is going to be about phylogeny. So phylogeny is the study of relationship in between taxa, and you can go two ways for morphological characters. You can either go for parsimony, which is like we'll select the shortest tree, which means that the tree that you would select that is considered to be the best tree fitting your data is the tree that has the less amount of evolutionary changes through your evolution step. Or you can go a little bit more mathematical ways with probabilities and Bayesian, which is based on the Bayes theorem that was developed during the 13th century, that you will calculate the probability of your tree, the probability of your data, and then you consider your posterior probability, which is the probability of your tree given the data that you have. The data being like your matrix, or your morphological matrix, etc. So while it was mainly used on biological data back in the 80s, mainly thanks to some algorithms that were developed during the 50s, we can now apply it thanks to the Lewis model developed in 2001 for morphological data. Right. So I'm just going to go through now all the results I've obtained on my PhD. And I'm going to start about ontogeny. My case studies were Biamoseuchians. So Biamoseuchians, it's a basal group of therapsids that thrive during the middle Permian. They go extinct before the PTB, and they are divided in two groups. So you have the basal-most Biamoseuchians that don't show any sign of ornamentation or bosses or ridge on the skulls. Then you have the Burnetiamorph that are considered to be little bone derived, that develop thick bosses on the eyes, on the side of the skulls, and also some ridges on the nose tail. Problem with Biamoseuchians, they're really, really rare into the fossil records. Okay, it's carnivorous, so that's a little bit normal that you find them less than herbivorous animal into the fossil record, but still. We only have about 20 monospecific genera in the world, which means even each genera is described based on one species. And usually the material you have for that species is only one, the holotype. So it really limits the possibility for studying ontogeny or intraspecific variation on those. So the problem is the first question I had during my PhD was what could be ontogenic characters on Biamoseuchian? If you would find a baby Biamoseuchian in the field, how they would look like? So in the past 30 years, there's been 3 small Biamoseuchians that have been found in the South African Karoo. You have 3 specimens only. And thanks to them, we were able to decipher and to show what a juvenile Biamoseuchian would look like. So those, compared to an adult specimen, here the juvenile on the left and the adult on the right, the juvenile will have bigger eyes compared to the size of the skull in comparison to the adult specimens. Juvenile specimens tend to not have the bosses or the ridges developed yet in comparison to the adult specimen. Then if you look at the bosses or the early stages of the bosses you will have on those juvenile specimens, what they have what they show on the CT scan is this radial vasculature patterns that are inside of the boss, which will indicate that the animal was still growing when it came to its death. While on the adult specimen in the bosses, it's really thick, it's really well formed. You don't see anything, and actually you don't see the sutures neither. While in the juvenile specimen, the sutures are very clear and you can follow them really easily on your CT scan. Another thing on juvenile specimens, they don't have a fully ossified brain case, while that brain case is nicely ossified on the adult specimen with thick bony walls. A really weird thing that I found on my specimens is that on the juveniles, the parietal had either 3 to 4 center of ossification. So you could see inside the parietal there was really distinct sutures inside, indicating that the parietal was still not well formed when the specimen died. And you had a similar morphology as well on the preparietal. One of the specimens strangely displayed the pattern with like 2 canine and a third one inside. So there was a series of like 2 replacement canine in the specimen. So remember in the introduction, I spoke about like those 2 canine patterns, showing that even some adult specimens in therapsids still have 2 canines, still have replacement canine. So this, like the 2 canine and the replacement canine pattern, is not really a sign of juvenile, but it could be a sign of the specimen being a juvenile when you put it together with like a non-ossified brain case, the absence of ornamentations, like sutures that you can see really well. So thanks to that, I was able, with like a very few specimens that I had, to draw like a basic chart for what would be an ontogenetic development in Permian. So the early stage, you will have like your specimen that would start with like the fusion of the pre-parietal, and then you will start to develop its cranial ornamentation. Then second stage, the specimen will fuse its parietal ossification, finish its parietal ossification. You will have a much more developed like cranial ornamentation to come to the adult stage where you have a great elongation of the snout with like a bigger allometry there and an important pachystosis development on the skull. That was for ontogeny. So that was just to show you how those therapsids, and in particular those Permian, were growing. I am now going to move to the taxonomical work of my PhD. I had the opportunity of studies like basal anomodonts. Anomodonts, they were a group of herbivorous therapsids that appeared during the Middle Permian, and they went through the PTB and the Triassic as well. There's 2 groups. There's one group that is considered to be more basal, that will show like a various of morphology with post-canine and everything, while the second group that you will call taxonomies, they are the more derived one, and they also show a morphology with like the absence of post-canine, the absence of like incisors as well, and they only have a tusk, and they would develop a beak as well. I had 4 specimens coming from there. So I had 2 iodocynodonts. One of them was problematic, this one. And I had like the holotype of another iodocynodonts. The problem with that specimen is that there was a few studies, like in the past 20 years, that suggest that that specimen in particular will not like be iodocynodonts here. And so it had to be like to go through a taxonomical rework. I choose to select like another iodocynodont, which is a type species, and patronomodon, which is like basal anomodonts, to compare it to the problematic specimens there. So thanks to the CT scan, I was able to go through the full description of inner skull anatomy of those animals. So we had like one of the specimen was displaying his orbitosphenoids and his parasphenoids, like one of the bones that you don't see often there in the descriptions. I was able also to fully describe the inner ear of Patronomodon with the vestibule and the floccular fossa. Those parts of the animal are really important because with linking them to the quadrate, which is like the articulation of the jaw, and the stapes, it's linked to the like hearing system. They didn't add any external ear yet, like mammals do have. They have an internal ear, so they will hear through vibration into their jaw that will be transmitted, signal will be transmitted through the stapes into the inner ear chamber. As a like anatomical comparison, I can see that the specimen, the 2913, that is a little bit problematic for taxonomy. It has post-canine teeth, like you would find in Patronomodont, but like you don't find those post-canine teeth in iodocynodont, or very few. Iodocynodont would have 1 or 2 really small post-canine teeth, not like you would have in the specimen. Another thing is like my specimens there didn't add any incisors. It's the same with like iodocynodont, but Patronomodont here have 1 incisors displayed. One of the main characters in the cynodont It's like the folded morphology that you would find on your squamosoles there at the back. So it's like nicely folded into 3D dimension, while the one in Patronomodon and in the one in the specimens that need to be reworked doesn't show any folded morphology. It's actually quite flat. Another distinctive characteristic is that my specimen had like what we call a dentary ball, I think. Okay, so through anatomical comparison work, where we could see that it's the problematic specimens there was closer in shape to Patronomodon than it is to Iodocynodont, although it's currently assigned to Iodocynodont. To go further into taxonomic reattribution, we went through a little bit of phylogenetic analysis, and we can see that our specimens there goes outside of Iodocynodont. All the Iodocynodonts are all forming a clade together, while our specimen is actually outside. It's like for like a in more basal position. So the conclusion of the paper that we had, it's like that specimen needs to be a new taxonomic unit, and we call it Nyapulia Dorseni. Okay, so that was a little bit of phylogeny, and now I'm just gonna go a little bit deeper into phylogeny, but not only on anomodont, but on the whole like clade of therapsids. Coming back to my introduction, I've showed you like all clade of therapsid, they appear during the Orson gap here, and we don't really have much information about what happened during the early evolution, which caused like a problems for phylogeny there. Some of the studies that went through phylogenetic analysis, they had conflicting results. Like, for example, this one will show that the Biamosuchians are the most basal with like Dinocephalians and Gorgonopsians, while another study from John Liu showed that actually those Biamosuchians might be branching with the Gorgonopsians and the Anomodon with the Dinocephalians. So there's a little bit of conflict of phylogeny. That could be due is like because our therapsid we evolved during a really short time, so about 10 million years. They went through a very rapid radiation, which possibly would infer a problematic polytomy at the base of the tree. It's either hard or soft polytomy, but in both cases, it makes like the resolution of the deep nodes of our phylogeny really, really complicated. So there's two solutions you can have there. So you can find specimens that would be contemporary to the Orson gaps. That's one of the first solutions. Well, in China, they have a really interesting like fossil fauna with Biceridens, Cynophonius, and Raranimus. Those three specimens where they were published in the 90s and in the 2010s, they were suggesting that they could be amongst like the most basal therapsids there, but like there was not really a geological work of the Danshagu fauna at the time. We didn't know like the datation of the geological layers. I had the opportunity of studying the scan of Raranimus back when it was published in 2010. It was suggested it could be like the basal most therapsids. So it's only a snout, unfortunately. So, but I was able to fully make a full representation in 3D of the nerve system into the maxilla, what we call the trigeminal nerve. And one of the branch of that trigeminal nerve has a morphology that is much closer to what you could find into pedicosal varanopsid, or like in comparison that you could find in therapsids. So thanks to that and other anatomical comparison, we could say in the paper that Raranimus has in fact a morphology that is like really basal into therapsids. And recent geological work of the area inferred that it could be possibly Rhodian, and which is like contemporary to the Orson gap. So, okay, cool, we fill the Orson gap. Second solution, while previous phylogeny all use parsimony and cladistic methods, there's other methods that have been developed since the 2000 for morphology with like more like adapted to morphological characters, which is called like Bayesian phylogenetic analysis. And in other groups, in fishes, in birds, and some lizards, it showed that it might be the method that could be a solution for like going through problems of polytomies at the base of evolution of some groups. So we're trying to do Bayesian phylogeny. I'm just gonna go through a little bit of math there. I'm sorry, but that's not too bad, I promise. So Bayesian phylogeny, it looks for the best tree according to the probability, to the posterior probability of the tree, given the data. The data can include your morphological matrix. It can include also some basic parameters that you can put in your model, etc. It can be used on morphological characters thanks to the model of Lewis developed in 2001, which allows you to incorporate into the model the branch length and the rate of substitution into your phylogenical model. The problem is like you will want to calculate the posterior probability of every tree that you have in your space. And that's basically impossible because there's an infinite number of trees that you can have, or like a very large number of trees. And the Bayesian would want you to do like the average of those all of those probability. And that's basically impossible to calculate. Even with like modern computers, you can't do that. So you have to go through what we call a random sampling walk, or what is called the Monte Carlo Markov chain process, which is also used in other part of mathematics and statistics. The Markov chain Monte Carlo, it's like a random walk into your parameter space. So you walk into your parameter space and you try to find the base value. So let's say you're new in town and you're trying to find the best coffee shop. But there's a large number of coffee shops. You can't all test them. So you will go to the first one, test the coffee. Okay, it's good, it's bad. Then you move to the second one, to the third one, to the fourth one. And doing so, you do like a really small sampling of all the coffees you have around you. But you can't test all the coffees. So when do you stop? When do you consider that you have the best coffee possible in your town? So that's why you have like to calculate a ratio of likelihood. Like you will calculate the likelihood of the best coffee that you have now and the likelihood of the best coffee that you would have into the next coffee shop. And based on that ratio, you decide yes or no. Do I move to the next coffee shop, or do I stay there? Do I consider that this is the best coffee around? Basically, the MCMCs of the Markov chain Monte Carlo does the same, but for your phylogenies. It will go to every tree, calculate the probabilities, calculate the ratios, and then decide, okay, now we have the best tree. This is the best possible that you can have, and I'm going to stop here and I'm just going to present you that tree. So the very convenient thing about Bayesian analysis, it's like you can add into your model some time and clock model. So you can, in my case, I use like the fossil buff death model, which is really well adapted for fossils data. And you can add into your model a whole bunch of parameters related to time. So you can have your speciation rate, you can have your extension rate, like the model parameters, the evolutionary rate, the time that you have, all of the time ranges of all your fossils. And that's allow you to produce a time calibrated tree. So in regards of the tree I've obtained for the therapsids, I found out that our therapsids, they appeared around 280 million years ago. So that's well before the Olson gap there. Then I showed that during that Olson gap, my tree suggests that there was a large number of radiation events and evolutionary events, suggesting that at the time, synapsids were really thriving. They were like diversifying, like everything, they were really happy there. In comparison, there was in contradiction, there were some studies in a few years ago that suggest that time of the Olson gap was an extinction time. While if it wasn't for what therapsids. They were really thriving and developing at the time. Another suggestion that I've obtained from my phylogenetic analysis is that the Piamosuchian would branch with the Dinocephalian and the Anomodonts would branch with the Pteriodonts. But the probabilities that I had at those nodes were really, really low. So it needs further work to be confirmed. Another interesting thing is while Perseridens from China, from Dashan Kufana, was previously suggested to be the basalmost anomodont, it actually branched on the tree with more closer to Garmosuchian and Dinocephalians. And another findings there, it's like Cynophonius, which was considered to be like also coming from China, which was considered to be on Enteosaurs. It actually branched at the base of like the Dinocephalians evolution, suggesting that Enteosaurs are actually paraphyletic. Another finding, which is similar to what Jennifer got in 2007, is that I found that the Pterocephalians were not really monophyletic and they're actually paraphyletic. So you will have your main Pterocephalians like Lycosuchus and Glanosuchus there, and then you have one Pterocephalian here that is Simorinella. It's actually branching outside the Pterocephalians and branch at the base of the Cynodonts evolution. So that has two applications. The first one is that Pterocephalian would be paraphyletic. And the second one, if it's confirmed later, it will feel like what we have about like the Gauss lineage of Cynodonts. Because Cynodonts, we don't find them really early into the fossil record. So there's a bit of like a Gauss lineage there. So that's what I'm gonna do now. That was PhD. And now, like for my postdoc, I've decided to keep going on that non-monophyletic problem of Pterocephalians. And next week, actually, I'm gonna scan like some specimens that we recently found during fieldwork into the Karoo that are really small specimens and that could be really affiliated to Pterocephalians, and they come from the Eotacynodontas phylogeny. I plan to compare them to other Pterocephalians we know from the time, and try to, based on the work I've done during my PhD on phylogeny, I plan to like incorporate them into my phylogeny and see if I can find another non-monophyletic problem for Pterocephalians with Cynodonts or not. I also plan to work a bit more with Russian colleagues. I got some scans from some Russian specimens, and I want to develop that collaboration as well. That was for PhD, master, and postdoc. And I want to thank my two supervisors, Julian and Bruce, for copying with me since 2016. And yeah, that's the end of the talk. Thank you for coming for my lunch talk. Do you have any questions?